Hi, hello, good evening to all. This is Murali PG going to have the class regarding with the discussion of Karnataka CET 2020 question paper. So, with respect to our Unacademy, we are going to have plus subscription. So last date for plus subscription is 15th of August. You will going to get the plus subscription in the value with respect to value 999, and that includes complete preparation through the detailed courses and special doubt classes, weekly quiz, etc. So you can subscribe through unacademy.com KCET. So here we are aiming towards Karnataka CET 2021. Use my referral code MuraliPG853057 during the subscription. Regarding with the plus subscription, okay. So this is the values, subscription values in the case of Unacademy for different courses with respect to the month. So month wise, 1 month, 3 months, 6 months, 12 months, 24 months. Here the price values are given. Okay. So by this brief introduction, now I will move on to KCET 2020 biology question paper discussion. Okay, my dear students, welcome to Unacademy. Here we are going to discuss with respect to biology question paper 2020 Karnataka CET. Okay, so let us have the discussion now. Okay, yes, we are going to start with the first question. Injection of an antidote against snake bite is an example of what? So in the case of snake bite, we are going to give, or we are administered with the injection of antidote against the snake bite is an example of which kind of immunity? Autoimmunity, innate immunity, active immunity, passive immunity. So in the case of this one, it is answer is D, that is passive immunity. So it's a passive kind of immunity where the prepared antibody is going to be administered with respect to that person, isn't it? Prepared antibody is going to be administered with respect to that person's body, and here the person is going to get immune power against that particular snake bite. Okay, so answer is D for the first question. Let us move on to the second question. Certain tumors are called malignant because of what reason? They are not neoplastic, they are confined to specific locations, they invade and damage surrounding tissues, they show contact inhibition. So in this regard, malignant tumor and benign tumor, there are two types, isn't it? There are two types. With respect to these two types, one is going to have damage to the entire body, that is the malignant. So they invade and damage surrounding tissues. So here this is going to have the problem in the case of human body. So answer is C, refers to they will invade and damage surrounding tissues. So it is spreading type of cancer to the entire body. So answer for the second question, it is C is the right direct actual answer. Okay, if you have any doubt, you can have the chatting. Okay, if you have any doubt, you can ask through chatting. Okay, that is the second question. Answer is C. Next question, question number 3. The transport of which neurotransmitter is inferred by cocaine or interfered by cocaine? So the transport of which neurotransmitter? So serotonin, acetylcholine, and dopamine and GABA. So here the answer is dopamine. So dopamine is one of the neurotransmitter which is interfered by cocaine. When the drug is going to be taken, in that person the neurotransmitter like dopamine is not working. So here the third question answer is A. Okay. The next question, question number 4. In the life cycle of plasmodium, fertilization takes place in which area? Whether it is in the liver cell of human being, salivary glands of mosquito, RBC of humans, or stomach of mosquito. Here the answer is D, refers to stomach of mosquito. It is also called as gut of mosquito. It is also called as gut of mosquito. Okay. So liver cells, salivary gland of mosquito, RBC of human being, and stomach of mosquito. And here that answer is D, refers to gut of mosquito or stomach of mosquito. Okay, where we can observe fertilization of that gametes. In the case of plasmodium life cycle will going to happen. So answer is D. Okay. Next question, question number 5. White rust resistant variety of brassica. White rust, it is one of the fungal disease. White rust resistant property is there in one of the variety of brassica, and which is that variety with respect to example wise. So it is answer is C, refers to Pusa swernin. Pusa swernin is showing white rust resistant quality. It is one of the brassica member. Okay, Pusa swernin, the name of that variety, Pusa swernin. So answer is C for fifth one. Okay. Next question. Which of the following plant tissues cannot be used as explant in tissue culture? So in tissue culture procedure, the cells are going to be used. So the living cells are going to be used. Here the answer is D, refers to Sclerenchyma, because Sclerenchyma is a dead mechanical tissue. So as explant in tissue culture, usually meristematic tissues are going to use it, and which are having the capacity to divide very fast. So continuously dividing cells. So regarding with this one, the plant tissues cannot be used is the Sclerenchyma under tissue culture methodology. So D is the answer for sixth question. Next, the seventh one. The next question. The hybridization between naturally incompatible plants like potato and tomato can be achieved through which method? So hybridization between naturally incompatible plants refers to belonging to different group. So potato and tomato can be achieved through which method? That is somatic hybridization. So somatic cells, vegetative cells, where somatic hybridization is a methodology to produce the hybrid variety of plant by mixing the character of tomato and potato. So it is called tomato and potato Pomato is the actual term used with respect to that plant, pomato. So that phenomena can be achieved through somatic hybridization. So somatic hybridization is one of the important technology in the field of our biological aspects. So to produce different thing. So in the previous question, one student asked what is GABA, isn't it? It is a gamma aminobutyric acid. Gamma aminobutyric acid. Okay, gamma aminobutyric acid. So answer for 7th one is C, somatic hybridization. Next question. A chili plant was severely infected with the Chilly Mosaic Virus. Identify the technique that helps to raise virus-free plant in the next generation from the above virus-infected plants. So one plant, it is here the chili plant. This was infected by Chilly Mosaic Virus, but we want that plant for our future generation. In that level, we can go for the tissue culture, isn't it? Tissue culture. So here it is the meristematic culture, tissue culture. Why? In this plant, if viral attack is there, but meristematic tissues are never infected with the virus. Even though the plant is having the virus content, meristematic tissues are not invaded with the virus. So meristem culture can be done, and as a result of that, we can get the next generation through the infected plant explant. Okay. So that is what the technique observed in the case of tissue culture, meristem culture. Okay, not artificial hydroponics or self-pollination. Here meristematic culture, because meristems are never infected by the virus, because the rate of cell division in the meristematic tissue is greater than that of multiplication speed of virus particle. Okay, so answer is C for the 8th question. Next, 9th question. Question number 9. Just a minute. Yeah. In sewage treatment, secondary treatment is considered highly significant because of what? So here it reduces the BOD of level of sewage. So in the secondary treatment, we are treating that sewage content by using anaerobic microorganism. So as a result of that, unwanted contents which are dissolved in the format of that sewage water is going to be taken up by that microorganism. So biological oxygen demand of that sewage water is going to reduce. So in the primary treatment, removal of debris like that and organic content, etc. But in the secondary treatment, where in the secondary treatment plant, we are going to treat that sewage water under anaerobic condition, so that reduce the BOD of water level. So answer for 9th question is C. Okay. Next, question number 10. Ruminant animals can digest cellulose in their food, whereas human beings are unable to do so. This is because of what reason? This is because of what reason? So here answer is A, because methanogens are absent in human gut. Methane producing bacteria, methanogens, anaerobic bacteria are present in the case of ruminant. If you take a cattle, etc., in their digestive system, we can observe methanogenic bacteria, methanogens, so they are able to digest the cellulose content of the food directly without any cooking. But in the case of human being, it is not possible, because we are not having methanogens in our human gut. So methanogens are absent in human gut refers to alimentary canal or digestive system. So the question number 10, that is A is the answer for question number 10. And next question number 11. From the given combinations of steps in PCR, identify the enzyme dependent step or steps. It is extinction only. Extinction refers to, here we are going PCR, polymerase chain reaction. It is important for the production of multiple copies of the same DNA in the case of recombinant DNA technology and other processes. So where during the extinction of that DNA molecule, the enzyme is very important. So identify the enzyme dependent step, that is the extinction step is going to have the utilization of maximum enzymes. Next question number 11. Biolistics method is suitable for gene transfer into which cells? Here as per the answer, it is the plant cell. Biolistic guns, or we can say it as gene gun, is going to be utilized for the introduction of gene directly into the plant cell. So answer for A is 12th question is A. Okay, plant is a plant cell. We can observe the utilization of that biolistic gun to introduce the gene. Next, identify the labels M and N in the following agarose gel electrophoresis representation. So in this one, in the labeled diagram, here M is what, N is what. So M is the largest DNA band, N is the smallest DNA band. So in agarose gel electrophoresis, here we can separate the DNA molecule according to their molecular weight. DNA is negatively charged particle. It shows movement towards a positively charged electrode, that is anode, according to the molecular size. Larger or bigger sized DNA strand will remain back, and smaller sized DNA molecule will move fast towards the anode side. So this is what the agarose gel electrophoresis, to separate the DNA strands with each other. So it is one of the important methodology utilized during the recombinant DNA technology to separate the DNA strand, etc. Okay, so answer is D for question number 13. Next, question number 14. Yes, in the 14th question, question, in RNA interface, the double-stranded DNA, dsRNA refers to double-stranded RNA molecule, prevents what? It is translation of mRNA. So in RNA interface, translation of mRNA is going to be prevented by the double-stranded RNA. So that is answer is D for 14th question. Next, 15th question. Nowadays, the early diagnosis of bacterial or viral infection in humans is possible using which technique? CT scan, serum analyzer, DNA sequencer, and PCR. So answer for this question D, that is PCR, isn't it? PCR can be utilized for early diagnosis of bacterial or viral infection. So in modern day or nowadays, in the case of human beings. So answer is D. PCR refers to polymerase chain reaction. Then which of the following features of plants is not hopeful in adapting to desert life? So here answer absence of trichomes on leaf surface. So here following features of plant is not hopeful in adapting to desert life is the absence of trichomes on leaf surface. Other things, presence of the cuticle, leaves modified into spines, presence of sunken stomata, these are helpful, isn't it, to adapt themselves to desert life. So answer is A, that is question number 16, A is the answer. Next 17th one. Here in the following equation of Wurl's per logistic growth, the letter R denotes what? So dn by dt is equal to Rn into K minus N by K. This is the equation according to the Wurl's per logistic growth curve, isn't it? So there are two growth curves. So one is this one, Wurl's per logistic growth, another one is exponential growth curve. So what is the value of R? Here what is the value of R? That is what? Intrinsic rate of natural increase. Intrinsic rate of natural increase with respect to that organism in the given locality, in the given time. So R is the value of intrinsic rate of natural increase. So answer is C. Next 18th question. The shape of the pyramid reflects the growth status of the population. Identify the type of age pyramid present below for human population. So answer is A. It is going to resemble this pyramid is going to resemble the declining population. Okay, it is not ascending or expanding or stable. It is a declining population. Here the less aged people. Here when you are moving upward, it is going to show the least percentage with respect to young age. So it is a declining population growth. Question number 9, A is the answer. 19, A is the, 18th question, A is the answer. For 19th question, here identify the possible link M in the following food chain. So in the food chain we can observe one M letter in between the plant, insect, snake and eagle. What is this M? M refers to, it is a frog, isn't it? It is a simple food chain. Plant or grasshopper insect and a snake, here frog, and next is a snake and eagle. So it is a food chain link in that M represented by the frog. So answer is D. Next, the organisms which invade a bare area to initiate an ecological succession are known as what? So the organism which invade, which cover a bare area to initiate an ecological succession are known as pioneer species. That is species of organism which will going to cover a bare area to initiate an ecological succession. So it is called the pioneer species, first formed species. So answer is A, pioneer species. Which one of the following is not included under in situ conservation? So here biosphere reserves, national park, biosphere reserves, national park, sanctuary, botanical garden. So in this one, in situ conservation, not included under in situ conservation, that is botanical garden. This not included under in situ conservation. It is the ex situ conservation, isn't it? Botanical garden is the ex situ conservation for plants. What about the biosphere reserves, national park, sanctuary? These are in situ conservation. Conservation of plant in their natural habitat. Conservation of plant away from its natural habitat under human control, that is botanical garden. So like that of zoological garden or zoo, here it is the botanical garden, one type of ex situ conservation methodology to conserve the endangered species or endemic species of plants. Okay, so answer is D. Next, 22nd. Which one of the following is a wrong statement? Wrong statement here. Ozone in upper part of the atmosphere is harmful to animals. This is the wrong statement, because ozone layer in upper part of the atmosphere is good to human animals or including human beings, because it will going to stop the entry of UV radiation from sunlight. Isn't it? So answer is A for 22nd question. According to Supreme Court of India, ruling with respect to Bharat Stage 6 norms, from which date these are supposed to be implemented in the country. So here, which is that date, that is B, 1st of April 2020 onwards, Bharat Stage 6 norms are going to be applied in our nation. So answer is B for question number 23. Then 24. Match the following classes of fungi with example. So in the column first they given fungi classes, in the second column they given the examples for that one. So answer is A, refers to, here what is that? Phycomyces, that is albugo. Second one ascomyces, that is penicillium. Third one is basidiomyces, that is nothing but Puccinia. And fourth one deuteromyces, that is Alternaria. So answer is A for 24th question. So match the following. So that is the fungi class and with the examples in the column second. See here, this is another classification. Observe the following simplified schema and choose the correct option that matches with the letters given in the boxes. So this is what actually the classification criteria of our vertebrata. So vertebrata is divided into two superclasses, two subdivisions and next superclasses. So subdivisions here, it is a lax jaw. Lax jaw refers to Ignatha. Answer is B here, Ignatha. And bear's jaws, so it is the Gnathostomata. And R, it is with the fins, that is the Pisces. Pisces are with fins, the swimming legs of the fish. And limbs are present in the tetrapod. So answer is B for the question number 25. So remember, here this is the classification of our vertebrata into Ignatha, Gnathostomata, then Pisces and tetrapoda. Pisces, that includes two groups, that is Chondrichthyes and Osteichthyes, and here tetrapoda, it is with the four classes, that is Amphibia, Reptilia, Caves and Mammalia. So answer for 25th, it is B. Next, 26th question. Here they given one floral diagram. So what is the labeled part? So in the floral diagram they given one label, that is what? What is this structure? It is what? Perianth. It is the perianth. The entire structure is the perianth. Individual unit is the tepal. And here it is unable to distinguish which is the calyx and corolla, isn't it? That condition is the perianth condition. And the sepals and petals are clearly visible if it is calyx and corolla. Calyx and corolla, it is fused, then is the, which is difficult to differentiate, then is the perianth. So answer is A for 26th question. 27th. A student observes grass and hibiscus plant in his garden during noon. To his surprise, only the leaves of grass were found rolled inward. The reason could be what? Why the leaves of the grass are folded in fashion? Answer is presence of bulliform cells in the grass leaves. So in the case of grass, we can observe presence of bulliform cells in the grass. So that is the answer. So bulliform cells are specialized cells observed in the monocot leaf in the case of grass. So we can observe rolling of that leaves. But in the case of hibiscus plant, the dicot plant, where bulliform cells are absent. Okay. Rolling of that one is because of the mechanism of this bulliform cells. So D is the answer for 27th. Next, 28th question. In the given diagram, identify the part which connects the peripheral microtubules to the central sheath of this diagram. These are the peripheral microtubules. This is the central sheath. What is the connection? It is the radial spokes, isn't it? It is the radial spokes of that structure. So that will connect our yes. That will going to connect the peripheral structure with respect to central sheath, isn't it? Peripheral microtubules to the central sheath. So this 9+2 arrangement is one of the important character with respect to cell organelle. So answer is A for 28. Next, 29th question. The element whose percentage weight is highest in both earth crust and human body is which one? It is highest both in human body and earth crust. It is the oxygen, isn't it? Not hydrogen, carbon, calcium. It is the oxygen. So answer is D for 29th. And 30th question. Identify the event in meiosis mediated by the enzyme recombinase. So meiosis, where the event in meiosis mediated by the enzyme recombinase, that is the crossing over. So crossing over is the change with respect to or exchange between a small set of genetic material between the non-sister chromatids of the homologous chromosome, crossing over, and this process was mediated by the enzyme recombinase. So recombinase is the enzyme that will going to have that phenomenon. Okay. So that is the answer here. So answer is a D for this question number 30. The deficiency of which of these elements interrupts photolysis of water during photosynthesis? During photosynthesis, photolysis of water is very important phenomenon. So photolysis of water, when the light passes onto this water molecule, one of the important step of the photosynthesis during light reaction, and it was interrupted by the following elements: Mn and manganese and chlorine. So Mn and Cl. So answer is B for 31st question. 32nd question. In C4 plants, C3 cycle takes place in which area? C4 plants, we can observe Crank's anatomy. So specialized modification is there, where the C3 cycle is happening or taking place inside the bundle sheath cell. So answer is A in the case of C4 plant. So here the Crank's anatomy is there. So bundle sheath cell, inside that one the C3 cycle is going to be happening. So if you take a C4 plant, it is the observation. So answer is A. But in C3 plant, it is not inside the bundle sheath cell. It's a normal happening. Next one. During citric acid cycle, During citric acid cycle, the various organic acid undergo decarboxylation. Of course, which of the following organic acids of the above cycle have 4 carbon, 5 carbon and 6 carbon respectively? So if you are writing the citric acid cycle, we can observe different intermediate compounds, that is 4 carbon compound, 5 carbon compound and 6 carbon compound. Here these compounds are succinic acid, alpha-ketoglutaric acid and citric acid. So succinic acid, it is a 4 carbon, alpha-ketoglutaric acid is a 5 carbon and citric acid is a 6 carbon compound. So answer is C for question number 33. Okay. Next, 34th question. See here, consider the following statements regarding photosynthesis and respiration in plants and select the correct option. Which is the right statement, which is the wrong statement. So answer is A, refers to statement 1 and 4 are correct. Rubisco has ribulose biphosphate, carboxylase, oxygenase. Rubisco has high affinity to oxygen in low carbon dioxide concentration. If high carbon dioxide concentration, then it will show the affinity towards carbon dioxide. So that is the correct statement. And next, fourth one, oxygen is a final hydrogen acceptor during aerobic respiration. These two are correct. 1 and 4 are correct. Then third one is wrong. Yeast poison themselves when the concentration of alcohol reaches to 7%. That is the wrong statement. Then this is not needed here. So answer is A for question number 34. Yes, now come to the match the following 35th question. So match the following items here. So here the pancreas, gastric glands, small intestine, salivary gland and their secretion, isn't it? Respective enzymes of the digestive glands. So answer is C, refers to the first one. Pancreas is going to produce trypsin. So in the form of chymotrypsin, inactive form, later it will become active. Then second is the gastric gland will going to produce pepsin in the form of pepsinogen, isn't it? That is for protein digestion. Trypsin is for protein digestion. Then third one, small intestine, that is Q enterokinase. Enterokinase is the activating enzyme. Then fourth one, salivary gland, very simple, tyalin, means salivary amylase. Another term for salivary amylase is the tyalin. Okay. So answer for the question number 35, it is C is the right answer. Next, question number 36. Once again, here they given match the different types of leukocytes, column 1 with their percentage of occurrence, column 2 in a healthy adult human, and choose the correct answer. Answer is A. So we know that neutrophils, lymphocytes, monocytes, basophils and eosinophil. These are 5 different types of WBC cells, isn't it? So out of these WBC cells, so answer is A. Neutrophils are with the how much? 60 to 65%, highest percentage. Then second one, lymphocyte, it is 20 to 25%. Third one, monocyte, here the answer is 6 to 8%. And fourth one, basophils, it is R 0.5 to 1%. And fifth one, eosinophil, it is 2 to 3%. This is the total percentage with respect to WBC cell. But when comparing with RBC, that is erythrocyte, it is very much lower. You see, 5 to 8,000 WBCs are there in front of 5 lakhs of RBC with respect to cubic milliliter of blood. Okay, so highest number is the RBC in the entire blood cells. Next is the platelets, and next is the WBC cells. So answer for 36, it is A is the right answer. Next, 37th question. In which part of the human brain corpora quadrigemina is located? That is the midbrain. So what is this corpora quadrigemina? It is the 4 round prominences structures which are present in the midbrain, and it is for the interpretation of result regarding with the vision and hearing. Okay, corpora quadrigemina. So answer is D for question number 37. Next, 38. A girl after attaining sexual maturity shows development of growing ovarian follicle, development of mammary glands, and high pitch of voice. These changes are attributed to dash hormones. So it is the estrogen hormone. Secondary sexual characters are going to be expressed during the puberty stage, and that is what estrogen going to show secondary sexual character in the case of woman, here in the girl, in the female, but in the case of male also we can observe secondary sexual character due to the male androgen hormones. Okay, so answer for 38, it is C is the right answer, estrogen. Next, question number 39. In apple, the chromosomal number of gamete is 17. Chromosome number of gamete. What about the chromosome number? What is the chromosome number in its primary endosperm nucleus? PEN. Answer is 51. Means A is the answer. So here gamete is N, whereas primary endosperm nucleus is 3N. So 3 times higher. So 17 into 3, that is 51. So primary endosperm nucleus is 3N condition, triploid. Gametes are haploid, haploid to triploid. So 3 times larger. So 17 into 3 refers to 51. Answer is A. Next, question number 40. Yes, here we given with one diagram. Identify the part called M here, which is that part M. Vegetative propagation is one of the asexual method of reproduction in the case of water hyacinth plant, terror of Bengal. And what is this structure? It is the offset. C is the answer, offset. So it is the offset between the two ends of between the end of this offset we can observe 2 plants. So it is one of the vegetative propagation where this offset is going to have the node and internode. So from that nodal region we can observe the development of new plants, and vigorous growth is there. So it is called terror of Bengal, and the structure is called offset. Okay, so answer is C for question number 40. Next, 41. During an excavation of soil, pollen fossils were retrieved from deepest layer of soil. The pollen grains remained as fossils because of what reason? So answer is here D, isn't it? So because the exine of pollen grain is highly resistant to enzyme action. So exine wall of pollen grain is highly resistant to enzyme action because what is the reason? Because the exine wall of pollen grain is made up of sporopollenin, organic matter. That sporopollenin is very strong in front of temperature, so that enzyme action and acidity. So acid condition. Till today there is no enzyme to degrade with respect to that sporopollenin. So sporopollenin is going to give full protection with respect to that pollen grain from damage. So remained as fossil for long duration of time because of the sporopollen observed in the external wall of pollen grain. So answer is D. Then 42nd question. Identify the mismatch. Mismatch one. Primary endosperm nucleus diploid, antipodals haploid, zygote diploid, synergies diploid. So answer is D. Why? Synergies are haploid. Synergies are the cells observed on the either side of the egg cell. Egg cell synergies are haploid. Antipodal cell is haploid. So synergies is haploid. The haploid ability. So it is the haploid. Instead of that, they given diploid. So answer for this one, it is D. 42nd. Then 43rd. 43rd. Yeah. 43rd question. Yes. Identify the correct order of events in pollen-pistil interaction from the given option. From the option given below. So what is the answer? Answer is C. The first of all, what happens? Deposition of pollen grain on stigma. Then entry of development of pollen tube. If pollen grain falls on the compatible type of stigma, stigma will going to allow the germination of pollen grain and development of pollen tube. Then entry of pollen tube into ovule, the next step. Through the pollen tube, which will carry that gamete nucleus into the region of ovule inside the ovary through style, isn't it? Then next is the entry of pollen tube into embryo sac, inside the embryo sac where egg cell is present. Then release of male gamete into embryo sac results in the fertilization, double fertilization in the case of angiosperms. So answer for the question number 43 is C. The sequence step of the double fertilization. Okay. The next point, next question. 44th question. Match the month listed in column 1 with the organogenesis of fetus in column 2. So here 1, 2, 3, 4, here A, B, C, D. First month, that is answer is A. In the first month, what happens? The heart is going to be developed. In the second month, it is limbs and digits. Fifth month, it is hairs on head, and sixth month, separation of eyelids. This is the gradual development of fetus inside the womb or uterus of the female reproductive system in the case of women. So this is the with respect to first trimester, second trimester, third trimester, we can say first trimester till 3 months, isn't it? Next second semester, trimester from 4th, 5th and 6th, and from 7th, 8th, 9th the next trimester. So it is given here like first month, second, 5th and 6th, okay, month. So answer is A for 44th question. Now 45th question, 45, question number 45. Yes, yeah. When the fallopian tube is blocked with respect to female reproductive system, at ampullary region, the ovum fails to move from which area? It is the infundibulum to isthmus. So ampulla isthmus. So answer is A. So ampulla isthmus portion is the site for fertilization. So when the fallopian tube is blocked at ampullary region, further movement of ovum, movement from infundibulum to isthmus is going to be blocked. So answer is A for the question number 45. Yes. Here they given oogenesis phenomena in the diagrammatic representation. Here we have to identify the cells represented as PQRS in the schematic representation of oogenesis shown below and choose the correct option. So with respect to this one, answer for 46 question, A is the answer, refers to P refers to oogonia, Q refers to primary oocyte, R refers to secondary oocyte and ovum. So here sequence steps are there under this oogenesis phenomena observed in the case of female reproductive system for the production of ovum. So answer is A for question number 46. 47. Which of the following contraceptives could be effective in avoiding pregnancy if used within 72 hours after casual unprotected intercourse or sexual action? So 47th question. Here the answer is A, refers to it is pills or the tablets which are with the progestogen estrogen combination. This is going to be taken within 72 hours after casual unprotected intercourse. If it is happened, then as a contraceptive method, under that method to avoid unwanted pregnancy, the woman can take the pill which is having the progestogen estrogen combination. So that is one of the important technique in the case of contraceptive methods. So answer is A for 47th question. Another few questions, 10 to 12 questions are there. Yes, 48. Choose the correct statement regarding gift gamete intrafallopian tube transfer or gamete intrafallopian transfer procedure. So correct statement is B, where ova collected from the female donor are transferred to the fallopian tube to facilitate zygote formation in the recipient woman or recipient. So this gift technique, it is coming under artificial method with respect to overcome the infertility, isn't it? Artificial ART, artificial reproductive technology. So here when the woman is unable to produce the ova is going to be collected from the donor, and that ova is directly introduced into the fallopian tube of the woman or to the recipient. So it is one of the technology to overcome infertility in the case of woman. So answer is B for 48th question. Next, 49th question. Yes. Which of the following characters was not studied by Mendel in his pea plant experiment? Answer is A. So here answer is A, refers to leaf shape. Apart from this A, all other things are going to be studied, or it is studied by this Mendel, isn't it? That is stem height, long or short, then pod shape, then seed shape. So all those things are studied. 7 contrasting character, isn't it? 7 important character with the contrasting features, but he didn't observe the character with respect to leaf shape, or he was not studied with respect to leaf shape of the garden plant under his experiment. So 49th answer is A. 50th question. Question number 50. Yes, yeah. In an organism, mutation in a single gene exhibits multiple phenotypic expression. Identify the underlying genetic mechanism in the above instance. So mutation in a single gene exhibit multiple phenotypic expression. So this answer for this one, it is B, pleiotrophy or pleiotrophism phenomena. So single gene will control multiple characters. Pleiotrophism phenomena. Pleiotrophy. So answer is B. Question number 50, answer is B. Next 51. A pure breeding pea plant with the round yellow seeds was crossed with the pea plant having wrinkled green seeds. On selfing of F1 hybrid of this cross, 64 progenies were obtained in F2 generation. Find out the number of F2 progenies showing non-parental characters. So non-parental character, it is 24. So we know that 9:3:3:1. That is for 16 outbreeds. Now it is 64. So if you take 64, then 36:12:12:4. Okay. Refers to 12:12 means non-parental character. So it is 24. And 4 is the parental character. 36 is the parental character. 9:3:3:1. In that 3:3 is the non-parental. 9 and 1 it is the parental. Now if you take 16 progenies, if you take 64 progenies, then it gives 24 is the right answer. Okay. So question number 51, answer is A. Next 52nd question. A man with blood group A marries a woman having blood group B. Here they didn't mention, they are not mentioned whether it is homozygous A or heterozygous A, B, homozygous B or heterozygous B. Generally they said the man with the blood group A marries with woman with blood group B. The maximum possible blood groups among their progenies are how many, or which are the blood groups? Answer is A, refers to A blood group, B blood group, AB blood group and O blood group. Maybe they, because if it is heterozygous A, IA, I, small letter I. Heterozygous B, IB, I. So I crosses with I refers to O blood group, isn't it? So A, B, AB and O. So answer is A option. Okay, is the right answer for 52nd question. (53) The length of DNA helix in a typical nucleosome is how many? What about the answer? B, isn't it? So if you take nucleosome model, the length of DNA helix in a typical nucleosome, it carries about 200 base pair. So answer is B for 53rd. Then 54th question. Which of the following types of RNA carries amino acids towards ribosome during the translation? So which of the following types of RNA carries amino acid towards ribosome during translation? So mRNA, rRNA, double-stranded RNA and tRNA. So answer is tRNA, refers to transfer RNA, which will transfer the required amino acid from the site of production to the site of protein synthesis, that is the ribosome. So mRNA is the messenger RNA. It is going to pass the message from DNA to RNA. That is the messenger mRNA production. rRNA is the structural component of ribosome. So answer is D. 54th answer is D. Transfer RNA, which will going to transfer or carry the amino acid required for the protein synthesis. So clover leaf model, tRNA. Okay. So 54, D is the right answer. Next 55th question. 55. Question number 55. Yes. In eukaryotes, the entire base sequence of a gene do not appear in mature RNA because of what? In eukaryotes, entire base sequence of a gene do not appear in mature RNA. Why? Because here introns are removed during the processing. So introns are going to be removed. Introns are not required. Only the exons are required. Exons are coding sequences. So non-coding sequences, introns are removed by using splicing phenomena under spliceosome action, and only the exons are going to be rejoined, and as a result of that, mature RNA is going to be appear. So it is happening in the eukaryotic processing level under the production of mRNA during transcription before translation, isn't it? But in the case of prokaryote, it is not there. So introns are removed during the processing, so exons are going to be there. So entire base sequence of the gene which is present in the DNA not appear in mature mRNA. Okay, so answer is D for 55th question. Then 56th question, another 4 to 5 questions. Okay, suppose DNA samples collected for DNA fingerprinting analysis are less than the required quantity. Which of the following techniques is useful to make the samples sufficient for above analysis? That is PCR, polymerase chain reaction. In the earlier question, another question I told that PCR is important for diagnosis purpose, isn't it? Polymerase chain reaction. So it is the third time it is there in our question paper, PCR word. So PCR is going to be utilized to obtain a large number of required copy of the DNA. So under DNA fingerprinting analysis. So same thing is there in the recombinant DNA technology also. So 56th question, D is the right answer. 57. When Escherichia coli means E. coli cells are cultured in a medium where lactose is absent, the I gene of lac operon continues to produce repressor mRNA because it is a constitutive gene. It is not structural, non-coding, operator gene concept. Here ABC is not right. D, a constitutive gene of that operon. So I gene of the lac operon continues to produce repressor mRNA. So answer is D. And 58. For the given sequence of DNA, identify the complementary sequence of bases on its mRNA from the option given below. So here I given DNA strand with 3' end, ATGCA, TGCA, TGC, 5' end. So for that, answer is B. So the opposite in the opposite strand, what is the nature of nitrogen base according to the base sequence rule? So it is second is B is the right answer. Okay, B is the right answer for 58. It is easy to write, isn't it? But K should be there. Their uracil will come instead of thymine, isn't it? Uracil will come instead of thymine against the adenine. Next, 59th question. Which among the following was the biggest land dinosaurs? So a direct question. Tyrannosaurus rex, biggest land dinosaur. Tyrannosaurus rex. So answer is C. And last question. In a population of plants, some were extremely tall and some remaining were extremely dwarf. No plants of the population showed intermediate height. The type of operation of natural selection in the above case is what? It is disruptive. It is disruptive type of selection. So answer is A for 60th question. So by this all the questions are observed under this Karnataka CET 2020 with respect to biology topic. So Unacademy is going to aim towards 2021 Karnataka CET aspirants. So you can subscribe for plus subscription. There you will going to gain access to hundreds of specially curated courses and batches of the best educators. So here you can join to this plus course, one who are there in front of 2021 Karnataka CET. So special introductory offer will be there till 15th of August. It is 1 month plan $9.99, and if you want to subscribe to that one, you just visit the website unacademy.com and use my referral code. So in the beginning I given that one here for the subscription. 1 month subscription plan is this one, and you can subscribe by using this particular website. Use my referral code MuraliPG853057 for the subscription, and this is the price value for that one. Along with biology you will going to get other subjects also. So 1 month plan 10% off is there within 15th of August. So this is the plans for different duration: 3 months, 6 months, 12 months and 24 months. So use this link bit.ly/igetCET to know more about the price values, and you can subscribe by using this link, that is unacademy.com. okay, Goal Karnataka CET, and use my referral code. So by this I am thanking you, all of you. So join Unacademy and fulfill your goal in the oncoming Karnataka CET 2021. So thank you one and all. By this I am going to end up this session here. Okay, thank you.